This is the first part in a video series that's going to be discussing bacterial pathogenesis. So in these videos, we're going to be talking about how do bacteria cause disease in humans. So the first thing we need to understand is they need to be attached, or in other words, adhere. The reason they need to attach or adhere is because they need to be able to colonize. So bacteria are going to attach through what we call adhesins. These adhesins include fimbriae, pili, and biofilms. Pili are not only used for attachment, but rather for conjugation as well. And conjugation is a way that plasmids are shared. Plasmids contain toxin production, which are going to be beneficial in a further video that we'll discuss. But I just want to show that pili are not just solely used for attachment. These are going to attach to host glycoproteins. Glyco means sugar, protein means protein. So these are proteins that are covered or coated with sugars. Then, once they attach and start to colonize, there are two methods through which they're going to further make sure that they aren't destroyed. That's invasion and evasion. So invasion is how a bacteria will get further into the body. So into an invasion. So that's how I memorize that one. Getting into the body is invasion. So the one method is called membrane ruffling. Membrane ruffling is where the bacteria is going to induce endocytosis and enter into a host cell. Our cells normally should not take bacteria in unless they're phagocytes and they're doing a different process. But normal cells should not just let bacteria enter into them. So that's why the bacteria induces endocytosis. There are also enzymes, but we're going to classify all the enzymes together. So this is your one invasion technique for right now. Then we have evasion. So you're evading, or in other words, avoiding the immune system or host defenses. Host defenses, such as the immune system, are going to try to eliminate microbes. One way is through a process called phagocytosis, which is where a phagocyte is going to engulf and destroy a bacteria. So they want to avoid that. So it's called anti-phagocytic structures. Anti meaning against, phagocytic standing for anti-phagocytosis, so against phagocytosis, and these are structures the bacteria makes to prevent themselves from being eaten. And these include capsules. So we talked about these with bacterial structures. These are sugary layers that are slippery, and also waxes. You don't need to know the structure of waxes, but you need to know one wax in particular, and that is mycolic acid. Mycolic acid is found with TB, tuberculosis. So tuberculosis prevents phagocytosis. You also can hide inside phagocytes. So the bacteria that can do this are pretty nasty because they have the ability to prevent themselves from being destroyed, and they're hiding inside the thing that's trying to destroy them. Imagine if you hide inside a serial killer's house, inside his garage. he thinks he killed you, and he's never going to look for you inside his garage, so you just hide out there. That's exactly what these bacteria are doing. They'll hide out, they'll continue to grow, eventually kill the phagocyte, and then be eaten again by another phagocyte, and then survive again. So these are pretty nasty bacteria. And then the last way is antigenic variation. Antigenic variation means it changes how it looks. An antigen is anything foreign to the body. Variation means changing, so this changes the foreign thing to the body. So the body recognizes it, creates memory against it. If you change that, now new memory has to be created, so it's harder to destroy it. So some microbes can change how they look. And then there's one more that isn't something that's discussed often, but I want to make mention, and that's mimicking host molecules. So if you look like the host is not going to attack you. So some bacteria know if they make the structures look similar enough to ours, they're going to be protected, and they can't have memory created against those. So these are your invasion and evasion tactics, and now we want to move on to the enzymes. So if you still need time to write down this material, I recommend you pause the video and do so now. Enzymes are proteins, and so bacteria can make certain proteins which are going to have special functions. And so enzymes are going to break down something or build something. So we have hyaluronidase, and this one is going to break down something. Specifically, it breaks down hyaluronic acid. So hyaluronic acid is found in the connective tissues. This is basically the glue that is holding your tissues together. So if this gets broken down, your tissues are going to fall apart. Necrotizing fasciitis, or the flesh-eating bacteria, produces hyaluronidase. So it doesn't actually eat your tissues or your flesh. It's just producing hyaluronidase, which is breaking down the hyaluronic acid, thus creating your body to fall apart and the tissues will begin to fall off. So we're also going to say this targets the connective tissue to help you remember where hyaluronidase is going to take effect. Then you have kinase and coagulates. These are both doing very similar functions, but they're just doing the opposite of each other. Kinase breaks down, coagulase builds. So kinase breaks down blood clots. So blood clots are formed in your body to act as a natural band-aid. The bacteria is breaking that band-aid down so it can further get into the body. Coagulase, on the other hand, is coagulating. It performs coagulation, which is where the blood begins to clot. So this forms blood clots to barricade itself. So you make the blood clot around you, the body now has to break down the blood clot before it can get to the bacteria. So it's a way of not being destroyed by producing some barriers. Then our last one is Iga Protease. Iga is what we call an antibody. So this one breaks down antibodies. Specifically, the antibody IgA. Antibodies attach to antigens. Antigens are anything foreign to the body. Obviously, a bacteria does not look like our body, unless it's doing the mimicking host molecules, but most do not. So bacteria don't look normal. So the antibodies are going to attach to the bacteria to flag it for destruction. Basically putting a red flag to say, hey, immune system, this isn't right, come destroy it. So a bacteria does not want to be destroyed. They want to be able to continue to colonize, grow, and not have to have those interruptions. Therefore, they create Iga Protease, which is going to break down those red flags in order to avoid the body from destroying it. Imagine you're in a dark room and you're trying to find another person. It's really hard, it's pitch black, you can't really see them. Eventually you might run into them, but how much easier is it to see them if someone put a glow stick on them? That person doesn't want to be caught, and so they produce something or create a way to get rid of that glow stick. That's exactly what's happening here. The bacteria are almost in essence in a pitch black room, so although you can find them and the body can find them, it's going to take a lot more time. These enzymes have kind of gray area. There's some ways to say, oh, it's for invasion, other ways to say for evasion. However, they do have the respective areas where they fall more in line with. So the top two are invasion tactics. So I put an I for invasion. So these help the bacteria get further into the body, while these two bottom enzymes are evasion tactics, or in other words, they're going to evade or avoid being destroyed by the immune system or host defenses. So this is the first video of bacterial pathogenesis. The next video we are going to discuss how bacteria are going to cause damage to our body.